Recording going. There we go. Okay, everyone. Hello, everyone. Welcome to today's webinar. Today, it's a pleasure to welcome Professor Lea Ann Dailey to talk to us about nano and microplastics. So Lea Ann is a pharmaceutical scientist by training with a research interest in inhaled medicines. She worked as a research scientist at Nectar Therapeutics, studying new formulations for inhaled antibiotics, before taking up academic positions at King's College London, Martin Luther University in Halle in Germany, and now she's at the University of Vienna. Her inhalation research has two major themes. So firstly, improving drug delivery to the lungs, and also understanding the toxicology of inhaled materials, including polymeric drug delivery vehicles. So the interest in polymer nanotoxicology is now being applied to the question of how inhaled nano and microplastics from the environment might impact lung health. So the title of the talk today is, are we inhaling nano and microplastics? And if so, what could be the potential effects? So with that, thank you very much for talking to us today, and the floor is yours.
Okay, well, thank you very, very much for the invitation. I'm really happy to see many of you virtually, and I'm also really happy to see many names in the list of people I know from my time in the UK. So it's a wonderful opportunity to at least say hi, hope everybody is well and that you're faring well under these somewhat challenging times. So I'm going to quickly get back to my presentation. Can I just have a quick confirmation that the...
Yep, we can see it, all looks good.
Okay, let me just start at the beginning then, and I am ready to go. Okay, so as in the brief introduction, I've actually come to the field of plastic inhalation from a tangential angle. As you heard, I'm a pharmaceutical scientist, and my original research questions had always been around whether biopolymers could be used via inhalation to control the release of drugs in the lung, and therefore change their pharmacokinetics in the lung. And one of the questions was always about whether we were introducing toxic materials in the form of polymers to the lungs if we wanted to use them for these particular ways. And somehow that has brought me into a space where I started finding some similarities between my own research and a quite current topic, and that is the prevalence of nano and microplastics in the atmosphere, whether we're breathing them and whether these can have some sort of health impacts. And I noticed that last week in the scheduling, you had Nick Molden talking to you about tire abrasion and tire wear particulates. And I thought, wow, that's actually really nice. You've probably heard a very nice introduction to this topic already. And so we are exposed through various sources to plastic materials that can be aerosolized, and they can be aerosolized in respirable size ranges. And these can come from numerous different sources. And we have both indoor and outdoor sources that could be potential ways of introducing these materials to our lungs. And I'm not going to spend too much time on this subject, but I did want to say tongue-in-cheek, I thought it was really funny that this week I'm sitting here in Austria, and all my colleagues in Austria are sending me now articles from the Daily Mail. And this article was in the Daily Mail, as well as a number of other different UK news sources, about another study that had been done where the numbers of respirable plastics were measured in different indoor environments, and a certain number of particles were counted, and therefore it was questioned in the public sphere as to whether inhaling different types of plastic particulates or polymeric particulates from different sources could be a threat to human health. And I think this is just one of many different different news stories that's come up in the past 2 to 3 years, that has really highlighted that it is a subject that people in the public are now really taking seriously. And I wanted to perhaps maybe remind many of you that actually almost 1 year ago today, you had Stephanie Wright from Imperial College London coming to talk to you about her work as a pioneer in this field, measuring atmospheric plastics. And I spoke to Stephanie actually right before this talk because I wanted to ask if she had any insight as to how these studies were carried out, because of course in news articles you don't get a lot of methodology information when it's published in the Daily Mail. And she said that she had some information that a very typical or common aerosol sampling method was used, where they used an active ambient air sampler with a vacuum pump. They ran the air through a filter, collected the particulates on the filter, and then you go through a typical sort of extraction procedure to try to extract the particulate material from the filter, concentrate it in a substrate. And then she said that her information sources indicated that these types of microplastics were, first of all, identified and then analyzed using a Raman spectral imaging method, which is one of the few methods that is now sort of at the forefront of being able to measure microplastics, especially of the size range that could be inhaled. And so I'd like to actually go back and remind you a little bit about Stephanie's work in this area, because she has done a lot of really careful analysis of an analytical method, basically, and it's not really a straightforward method, and therefore it bears a lot of scrutiny because we simply haven't refined the analytical techniques to be able to identify plastics amongst other types of particulate matter in many different environmental matrices. From this particular paper that Stephanie has published, you often will put the sample onto a substrate and first of all do an optical screen to try to visually identify particulates that look like they might come from a plastic or have plastic associated with this. And from this visual information or microscopic information, you can get both the size and the shape of the particulate. And these visual methods have to actually be combined with some sort of chemometric analysis in order to be able to identify which plastic material is there and whether it is a plastic or not. And two methods at the moment have been used quite regularly: Raman spectral imaging and FTIR imaging. Both of these methods have the limitation that the lowest limit of detection or resolution, where you get a high enough peak to be able to identify the plastic material, tends to be around the upper limit of respirable particulates. So with Raman spectral imaging, it's about around 5 micrometers in size. And with the FTIR imaging, a lot of sources are saying that it's about 10 to 11 micrometers in size. And as we all know, respirable material, the bulk mass of it tends to deposit deeper into the lungs, the smaller the aerodynamic diameter. And usually aerodynamic diameters well below this size range tend to be the more important. So I wanted to maybe, after discussing this study very quickly, or this news report very quickly with Stephanie, I wanted to go back and talk about this number. Up to 7,000 particles are breathed in from clothes, toys, furnishings every day. So we have a particle number per day. And at the moment, I don't think we really have enough information to understand what these numbers mean, because currently, due to some of the limitations with the analytical techniques, it's difficult to understand or have a really good feeling for what the actual exposure is in different environments, and how the inhalation of this particular particle number, where we don't know what the size, what the composition, what the surface chemistry is, and how that impacts our health, our lung health specifically. So I wanted to maybe just briefly show how difficult it is in terms of detecting and interpreting the data you get from some of these Raman spectral imaging or the FTIR imaging. And this is just a picture from Stephanie's study a year ago, just to show how the setting of the image analysis parameters really affects our ability to actually gather unbiased data and also reproducible data. So this is just to show you what the signal looks like if you have standard polystyrene particles of different sizes. So Stephanie took 10 microns, 4 microns, 2 microns, and 1 micron polystyrene particles, monodisperse, spherical, well-known composition. And these are the types of spectral images that you see. And they are separated into the wave numbers that are representative for this particular polymeric material. And I think what Stephanie shows here really nicely is that using different image analysis methods, you actually get different sensitivities. And I took some of the reported success rates of the different image analysis processes just to show that when you've got larger particles here, you get relatively high success rates, no matter what type of image processing method you use. But as the particle size gets smaller and smaller, even the most sophisticated image analysis processes still decrease in terms of their success rate of being able to successfully, correctly identify a polymeric particle within these critical size ranges that are respirable. So I think that was just my quick introduction to show you that the state of the art at the moment of detecting plastics in the environments that we might be breathing in is still at the moment very much centered on improving the sensitivity, the reliability, and the automation of many of these detection methods. It's difficult when we have very well-defined samples, and it becomes even more difficult once we have very complex samples. And there was another news report that came out around the same time that I thought was also very interesting. That's this one. This study was actually performed right here locally in Austria, where at the top of the Alps, at the Sonnenblick Observatory, snow samples were being taken to look for different types of organic matter within the surface snow. And the researchers, by chance, happened to see a diverse range of nanoplastics. So I wanted to quickly highlight this study as well, because it shows a very different methodology that's also being used, and shows the different types of approaches that different researchers are taking in this field. So in this case, the researchers took 2 centimeters of surface snow. They put it in containers, they let it melt, and then they filtered these samples through Teflon filters with a defined pore size of 200 nanometers. So what they're doing here is they're pre-selecting the sample so that you have a physical separation of particulates below or above this cutoff range. And then they're taking a mass spec analysis method to actually determine the mass concentration and the type of plastic within the sample. What I liked to see in this study is that they had numerous blanks that they took on many different steps of the process to be able to control for background plastic contamination. So that's a really big plus. What you don't get, though, from these mass spec-based methods are size information. So the only size information you have is that the materials that you're measuring in these samples have to be smaller than the filter pore size of 200 nanometers. And I thought it might be interesting to share what they saw. So what kinds of plastics are depositing up on the top of the Alps? Interestingly enough, they saw that polyethylene terephthalate is the predominant plastic. They took daily samples over 2 months from February to March in 2020, but they did see that also polypropylene made up a large portion of these materials. So that's what you're seeing, a concentration-based description of the different types of plastic materials that they saw. They also looked at weather conditions and precipitation conditions, and at least from my naive points of view, because I'm not a meteorologist or environmental scientist, I thought it was very interesting to look at the correlation that they found between the type of plastic. So here we've got PET concentrations and the correlation with precipitation, and they found the highest concentrations of PET on days where there was no precipitation at all. There could be many different explanations for this type of plastic transport. It was also interesting to observe that they saw no correlations between precipitation conditions for the polypropylene. And they were discussing whether maybe different types of plastics might do, depending on their different characteristics, such as density, have different modes of transport. Meteorologically, excuse me. So now I'm going to move from just 2 examples of some of the many emerging studies where different groups internationally are trying to understand how much plastic is in the atmosphere, how is it transported, where is it landing, and is it available in respirable amounts? And I'd like to move now to studies and some of our own studies about how plastics may interact in the lung. There are various different studies looking at different deposition profiles of aerosols, dependent, of course, on mass, density, number concentration. And one area that has also been of a huge interest is trying to understand how monodispersed nanoparticles deposit in the different regions of the lung. I chose one study actually from 2018 to quickly highlight here, because I thought it was interesting, because here actually a group in Sweden was actually aerosolizing polystyrene nanoparticles of different size ranges and asking volunteers, healthy volunteers, asymptomatic smokers, and COPD patients to inhale these. And they were using basically a technique where they had the patients inhale the aerosol or aerosolized nano particles from a reservoir. And in this particular study design, they actually had the patients then hold their breath for 10 seconds. This was allowing the different sized nanomaterials actually to undergo diffusion whilst being retained in the lung to understand where the regional deposition might be. And then they were exhaling. And so then characterizing the amount of nanoparticles in the exhaled breath and comparing that to the amount that was delivered with the inhaled breath. So they were trying to determine how much nanoparticle deposition was occurring within one breath. And they were delivering 3,000 to 6,000 polystyrene particles of this size per centimeter air. Cubic centimeter air. So I thought this particle number was very interesting because it might allow us to do some back of the envelope calculations about exposure surface area of these particles later on. And what did they see? They saw something that's very, very similar to many other studies with both modeling nanoparticle deposition in the lungs as well as other types of measurement techniques. They saw that there was a high level of retention of these small nanoparticles in the lung. The smaller 50 nanometer size particulates were retained to higher extent than the larger size, and this is because of the higher diffusivity of the smaller particles. And they didn't really see differences in regional deposition. Also, what they saw, which has been seen in other similar studies, is that COPD patients had lower deposition rates of these nanoparticles in the lung, very much likely due to the low levels of diffusivity of the air and air mixing in COPD patients with extensive emphysema, and therefore low mixing of air. And I just thought it was perhaps interesting to talk about now when we're trying to link how much nanoplastic we might be inhaling from the environment, and now seeing studies where actual nanoplastics and not other types of materials have been used for inhalation deposition studies. So I'd like to now go back in my research career to a place where I started out early on, and why I started looking at the toxicology of inhaled polymers. My PhD was all about taking biodegradable nanoparticles, putting drugs in them, making nanoparticles out of them. And the idea was we were going to nebulize them for patients with lung disease, and then see if they can slow down the release of the drug in the lung, and therefore extend the duration of efficacy of those drugs. And in the middle of my PhD, I started reading the literature from studies in occupational health sectors and air pollution sectors, and saw that a variety of different types of nanomaterials, when inhaled, can cause pronounced oxidative stress, acute inflammation. This can turn into a chronic inflammation cycle with impaired lung function. In some cases, this can induce neoplastic transformations. And what is now really well known is that this sort of inflammation can spill over to the cardiovascular system, and inhalation of air pollution is certainly very much associated with cardiovascular morbidity and mortality rates. And so I was thinking, well, could these biodegradable nanomaterials cause the same sort of side effects that other types of nanoparticles cause in the lung? And so one of the very first studies we looked at here was a comparison between biodegradable polyester nanoparticles with polystyrene, which is one of the common model materials of inert nanomaterials or polymeric nanomaterials that has been used as a standard for many, many years now. And so we instilled a certain number of nanomaterials, or a certain dose level of these nanomaterials into the lungs of mice. At the time, we were looking at similar mass concentrations, and then we looked very, very simply at different responses of inflammation, acute inflammation, and neutrophilic inflammation was one of the primary responses that we had seen in the literature. And we saw that the polystyrene nanomaterials, the non-biodegradable materials, were inducing a much more pronounced pro-inflammatory effect 24 hours after installation compared to our materials as well. And so this was a very simple study at the time, probably not very, very well thought through, but it gave us the indication that maybe polymers differ amongst themselves in terms of their surface properties, and therefore we decided that we wanted to try to look into how maybe polymers as a group are different from each other. And so later on, several years later, we started to play around with the idea that already in the field of nanotoxicology, many people had been looking at the size, the diameter of nanomaterials as a predictive factor of pro-inflammatory response, but really it seemed to be surface area driven. So of course, the same mass of smaller particles will have a higher surface area than the same mass of larger particles. But also the surface reactivity was a very, very important component, and many studies were showing that surface charge was also important in terms of either promoting or dampening inflammation. very specifically highly cationic surfaces and even highly anionic surfaces and surface charge density seem to play an important role in this compatibility or lack of toxicity or response. And of course, then biopersistence is a big factor. So in the past work, we had looked at non-biodegradable materials versus biodegradable materials. Biodegradable materials can degrade over shorter or longer periods of time, and we didn't know how long the biopersistence window is. Is that important in terms of whether toxic response will occur, especially if you're inhaling these materials on a regular basis. But the one factor that hadn't really been quantitatively measured in response to pro-inflammatory outcomes was surface hydrophobicity. And in comparison to many metal oxide particles or inorganic particles, polymeric particles have a more or less strongly hydrophobic surface, and this surface can attract many different molecules from the environment or from the physiological compartment that they're in. And at that time, at least, there wasn't really a good quantitative method to be able to characterize surface hydrophobicity. So that was one of the big questions that we wanted to address with some of our studies. Can you create a panel of nanomaterials with different surface hydrophobicities and understand if hydrophobicity plays a role in the local tolerability in the lung? So I've got a number of differently colored spheres here, and I'd like to highlight a select few of these spheres. So I'd like you to pay attention to the green sphere, the orange sphere, and the red sphere, because those are our polymeric materials that we were using in these studies. We used polystyrene as a common model so that we would be able to compare our results to other results in the literature. But we were also working with a polymer called polyvinyl acetate. Polyvinyl acetate is interesting for the reason that it has two different distinct chemical moieties as side groups. It has an ester group, which is very hydrophobic. That's the acetate part. And then under different chemical conditions, you can saponify the acetate and turn it into an hydroxyl group, and that gives it a hydrophilic component. And through different reaction conditions, we were able to create polymers that had a higher number of hydrophilic groups. That was our PVA 60%. So it was 60% hydrophobic and 40% hydrophilic groups. And then PVA 80, I have a typing error here. That should be PVAC 80 because we had 80% hydrophobic groups and 20% hydrophilic groups. And through differentiation of these hydrophobic hydrophilic group ratios, we were able to create materials that inherently had different hydrophobicities. And we also designed a method to actually quantify and score the hydrophobicity of each of our materials. So we used hydrophobic interaction chromatography. We ran the particles through there and looked at particle retention times through a number of different columns, and we gave them the score. And the higher the score was, the closest to 1, the score was, the more hydrophobic the score was, or the particle surface was. And so as you can see, polystyrene here in red, very, very hydrophobic. Our polyvinyl acetate 80%, also a high surface hydrophobicity, and the polyvinyl acetate 60%, much more moderate hydrophobicity. And then we had a couple of other types of particles of different materials, such as lipid-based systems or protein-based systems that were of interest to us at the time, but you don't have to worry about in this talk. And after that, we performed a similar study again in mice. This time we tried to improve our study design a bit. Instead of administering the nanoparticles as a bolus suspension. We actually tried to aerosolize the materials into the lung as a coarse aerosol to try to improve the deposition homogeneity. And then we also used 2 doses, and these doses were surface area based on a theoretical surface calculation. So we used 22 centimeters squared per lung dose and a 220 centimeters squared per lung dose. And I'd like to maybe perhaps highlight at the moment here that at this point, these concentrations did not necessarily represent concentrations that would be relevant for plastics inhalation from the environment. They're much, much too high. These were concentrations that we were thinking might be relevant for therapeutic administration of particulates, and so therefore they are at much higher doses. We then looked at inflammatory outcomes 24 hours after dosing, and supporting our hypothesis that hydrophobicity of the material surface does make a difference. We did see that the polystyrene and the polyvinyl acetate hydrophobic particles really caused a lot of acute inflammatory response, and this was dose dependent. And the more hydrophilic materials at both doses did not seem to show any inflammatory response. But what we also saw that was very interesting to us at the time, which we still don't understand fully, is that the polymeric materials in our systems had some interesting effects on the alveolar macrophage population of the lungs. And so I've got 2 pictures here that show just H&E staining of the lavage macrophages that we took from the animals, with the normal macrophages up the top. And what we saw after exposure to polymer nanoparticles in particular were these very highly vacuolated macrophages in our lavage fluid. And so we decided to just count the number of macrophages that showed this really highly vacuolated phenotype. And what was very interesting to see is that although the hydrophilic nanomaterials showed no inflammation, they showed that almost up to 50 to 60% of the entire number of macrophages within this lavage sample seemed to have this highly vacuolarized phenotype. Now, because these are nanoparticles, we know that the light white spots that you see in the pictures are not single particles. These seem to be some sort of intracellular vesicle that was developing after exposure or perhaps uptake of the particles within the cells. Interestingly, we saw that this was occurring with the hydrophobic particles that were inflammatory, but to a lesser extent. And one of the perhaps explanations for this was that we did have some indication that the more hydrophobic materials were aggregating once they were administered into the lung, and may not have been taken up to the same extent as the hydrophilic particles that didn't aggregate that much. And so one of our questions was whether perhaps uptake of these particles within these macrophages was doing something intracellularly that was leading to this phenotype. In a later study with a different strain of mouse, we wanted to ask the question of whether these macrophage phenotypes disappear after a single delivery, or do they persist over time? How long do these macrophages hang around and stay like this? So we dosed animals again with a similar surface area dose, and we looked at the lavage fluid 1 day, 1 week, and 1 month after exposing these animals to these polymeric particles. We used silica nanoparticles in this case as a control, because we're looking at a number of other types of toxicology outcomes, such as fibrosis development as well as inflammation. And what we saw was pretty much the same thing we'd seen in earlier studies. The hydrophilic particles did not cause any inflammation. The hydrophobic particles caused a pronounced inflammation, which resolved after day 1 and wasn't seen again at day 7 and day 28. But in both cases, we still saw these vacuolated macrophages. Their numbers were quite prevalent a week after. The prevalence of these macrophage cells within the lavage after 28 days seemed to go down, but did not disappear entirely. And so now we are still interested in trying to understand how these polymeric nanoparticles might influence these macrophages, not only in terms of their perhaps signaling pathways, in terms of perhaps signaling that there is particulate material here that needs to be removed, but also whether macrophages that have this interesting phenotype are still able to react in a normal way. Can they respond to other types of stimulus, such as bacteria? Do they still have their full host defense function? We didn't see any indications that these macrophages were apoptotic or dying. They didn't seem to be inflammatory or active in an inflammatory way. And they were still also, interestingly enough, able to take up other types of polymer nanoparticles. So they are somehow still functional, but the full impact of this type of phenotype is still unknown. And finally, I'd like to just present one final study that we did at King's College. This study was led by Ian Mudway, and he, along with other collaborators in our group, looked at incubating the hydrophilic version of the polyacetate nanomaterials in human epithelial lining fluid that was extracted from lavage. And we wanted to understand which proteins from the human proteome in the lung actually adhere to the surface and build a protein corona on the particle surface. And I haven't gone to a lot of detail from this study, but one of the really interesting and quite surprising results from this work was the number of different proteins that we actually found tightly adhered to the particle surface. So we compared our polymeric particles with silica particles. And with silica particles, you typically saw about 430 different types of proteins that were bound to the surface. 400 of these proteins were also bound to the surface of the polyvinyl acetate particles. But in addition to that, we found another 300. And so just in terms of the diversity of proteins that can be tightly bound to polymeric surfaces and compared to inorganic nanoparticles is vastly different. And we also saw different types or different prevalences of different proteins which were attached to the surface. And I'm sorry that I forgot to put the link here to the publication, but I can definitely provide that if you're interested in that. And so that leads me now sort of to the future of our work, because it is well known that pristine plastics can definitely provide a hydrophobic surface onto which many different types of biomolecules will adhere. And this will happen in the environment. The ability of these plastics to interact with other molecules will change over time and due to weathering processes, and that is something that is being investigated. But at the same time, it's quite important to just realize that the plastics will take up molecules from the environment. And one question that was addressed by some collaborators of mine is whether different types of plastic from the environment can specifically collect allergens from different sources onto the particle surface. Now, I want to just quickly state that most of my colleagues interested in this topic are working in the area of food allergy. And so they're very much working on the question of whether plastic accumulation in shellfish, for example, can specifically enrich allergens on the surface and therefore perhaps influence allergic response in people with shellfish allergies. So they got together and they started putting a grant proposal together to try to understand how allergy and allergic disease might be affected by microplastics in the environment. They also decided that they wanted to look at the effects of inhaled plastics and see if plastics may have the theoretical ability to interact with different types of pollen allergens and therefore influence respiratory allergies as well. And so these ideas have come together in a Horizon 2020 project that was just recently funded under the name of Imp Talks. And Imp Talks is basically looking at the question of whether nano and microplastics in the environment can impact human allergy and allergic disease. As I mentioned, the main focus in this is looking at food allergy and as well as whether plastics enrich in soils that contain high levels of plastics or irrigated with plastic containing water. And this exposure scenario then is very much focused on the oral ingestion routes and whether plastics with allergens accumulated and enriched on the surface can change the way they interact with the gastrointestinal system. And we have a small respiratory arm in this project that I'm involved in, where we're going to be looking to see whether plastics may be inhaled and theoretically whether interactions with different allergen proteins could impact then respiratory allergic diseases such as asthma. I fortunately don't have any data to show you from this at the moment. My big role in this particular project is actually to make model plastics. For this, we're using a lot of our knowledge from basically medicines development. So we're using tools from pharmaceutical technology to mill plastics, to define them, to characterize them, to create them in a sterile environment without endotoxin contaminants, and so that our collaborators are able to look at plastic interactions with very specific cargoes such as allergens. So at the moment, what I'm doing is I'm making a lot of plastics in my lab. I'm hoping or trying to make sure that my PhD students and colleagues who are making them are not inhaling them while they're making them, and so that we don't have any plastic toxicity as an occupational health risk with us. Before I finish this talk, though, I'd like to also maybe just tell you a little bit about some of the new colleagues that I've met since I've been here at Vienna. I've only been in Vienna for 2 years now, and of course most of that has been under lockdown. But recently I found out that there is a very vibrant collaboration with aerosol scientists and a huge interest in aerosol physics here in our university. And I don't know if some of these people are already known collaborators or known to you, but I thought possibly they may be of interest as maybe somebody who might be a future speaker in your seminar series. And I'd like to mention two people. That's Bernadette Weinziel, and she's basically an aerosol physicist, and she's been developing a lot of new and interesting impaction devices, which she takes up into airplanes and collects a lot of samples from the upper layers of the atmosphere, and then has been analyzing these. And in the course of some of these studies, she has also been measuring plastics. And so therefore, she's also just recently headed an application for a doctoral training center here in Vienna on this topic of atmospheric plastics. And she's been working together with Andreas Stoll, who is in the meteorology department. And he's been one of the people who developed the FlexPart modeling system, which I'm learning a lot about at the moment. And so he's working together with Bernadette to try to measure plastics in different locations and then model their transport based on their aerosol properties. And so there are a lot of activities ongoing in this area. We've also got a large number of collaborators from chemistry, physics, instrument development as well. So if there is further interest in this topic, or anybody would like to perhaps collaborate, I think we've got now a critical mass of people looking at these aspects from very different perspectives. And so for me, it's an exciting time to be in Vienna. I just wanted to thank a lot of people. Mostly this work was all done at King's College, and that's actually a few years ago before I left. As I said, after my move, we weren't doing a lot of work in this area, but now we're really happy to get back into the fields. And so I'd like to thank my collaborators, and thank you for your attention. And I'd be very happy to answer any questions that I can.
Thanks, Lea Ann. That was a fascinating talk. So as usual, if you have any questions that you'd like to ask, please feel free to unmute your microphone and pose them directly to our speaker, or put your questions in the chat. So any questions? So we have a question from Pete, and he asks, do we have any analysis on the nanoplastic composition in different environments and the hydrophobic, hydrophilic properties, e.g. tire wear?
So tire wear is definitely not my specialty, and that would be a very good question for your previous speaker from last week. What I do know specifically about tire wear is that there has been some postulation that specifically nanomaterials might be generated through vaporization during high abrasion effects in breaking, and then recondensation. But very, very quickly, most of the nanoparticulate matter heteroaggregates with lots of different materials in the environment, so they can be extremely diverse in terms of composition. With regard to other sources, especially in the nano material field, it's not quite so easy to address. So I think the snow study up on the top of the Alps was really interesting from the point of view that they had a relatively pristine environment with the surface snow, and they found material there that they could say had these distinct different types of plastics in it. But what they couldn't address is whether these plastics were somehow heteroaggregated or within a single particle or not. Were they different particles? How big were these particles? All they could say was that they were smaller than 200 nanometers. So it becomes very difficult, because the nanomaterials in particular you can't visualize. You have to look at the composition indirectly.
Yeah. We have another question from T. Tetley. I might be Terry Tetley.
Hi, Terry.
Regarding the foamy macrophages which occur in humans exposed to, for example, asbestos, it can sometimes be related to surfactant clearance and altered lipid metabolism. Did you look into this?
Yes, so we haven't looked into it as closely as we should have. So sometimes the altered lipid metabolism can be visualized in electron micrograph images. So you can see these lipid inclusion bodies. We definitely did not see that. And we did not see basically a high neutral lipid stain, which is also a second way of seeing whether you've got lipid accumulations. So we don't actually think that, I think possibly the lipid metabolism could be a bit altered, but I don't think you have a classical phospholipidosis in this case. So I would say we would like to look at this more, but we definitely don't see the lipid inclusion bodies that you would see in other types of foamy macrophage phenotypes. And that's why I'm a little bit afraid to call it foamy macrophage, because they're vacuolated, but we're not quite sure why. Some people have postulated that it could be a result of a dysfunctional autophagy response, and we tried to look at autophagy markers, but we haven't gotten very far. We didn't get really clear results on that.
Great. Any other questions for Lea Ann?
So I apologize, it was very biological in in the end.
We have a good mix of people on the CDT, lots with very strong biology backgrounds. I had a question which was to do with the internal mixing state of aerosols. So do we have any idea as to how important, for example, you've got these microplastics, if they're internally mixed with other insoluble components? So for example, it could be carbonaceous particles, so things like carbon particulates or metals, those kinds of things. If those encapsulate plastics, and therefore that plastic surface isn't exposed to.
What's been interesting is many people in the fields of engineered nanomaterials wanted to do it the other way around. They wanted to encapsulate the metal nanoparticles with the plastics. Because the plastics are definitely much more inert in terms of their surface. I haven't seen the other version where you're trying to protect people from plastic exposure by some type of encapsulation in that way. I think there had been, and I'm afraid I don't know the exact results, there have been some studies where people were looking at mixes of inorganic materials with plastics and looking to see if, or for example, cigarette smoke plastics at the same time, to see if the disease presentation, when you have a number of different stimuli, changes. And I'm afraid I don't remember what the outcomes were, if the plastics actually enhanced then the response to tobacco.
Yeah.
Thanks.
Maybe somebody else knows that question, actually.
Badio Zaman, have you got a question? No, he's muted. Okay, any other questions? Well, thank you very much for those webinar speaker recommendations. I've made a note of those two names, so we'll definitely stick that on our long list, and hopefully we'll get around to inviting them. And if anyone does have any ideas for who they would like to suggest as speakers in the future, then please get in touch. Right, well, there's no more questions in the chat, so I think with that, we'll end today's webinar and end by saying a big thank you to you, Lea Ann, for taking the time to speak to us today. So yeah, thank you very much, and we hope it all goes well in Vienna. I suppose still developing your lab.
Yeah. But thank you, everybody, for taking the time today. I appreciate it.
Thank you very much. Okay.